There are two ways for organisms to harness energy from their environment, one using chemical compounds and the other using the sun. This energy is in the form of adenosine triphosphate, or ATP, to perform work in cells to uphold life. It is important to understand that this base, adenine, and 5-carbon sugar, along with 3 phosphate groups, does not represent energy, but contains it in its chemical bonds. With this being said, if a phosphate group breaks off, making a new structure, a ribose, adenine, and 2 phosphate groups, it is known to be adenosine diphosphate, or ADP. The breaking off of this phosphate is the energy that is released, allowing the cell to do work. The addition or subtraction of phosphate is fairly critical in the processes that we're going to be discussing. ATP is needed in both prokaryotic and eukaryotic cells. Metabolism has many chemical reactions that convert molecules into other molecules and transfer energy. This is an important concept to understand because metabolism is divided into 2 branches: catabolism, which is the breakdown of molecules to produce ATP, and anabolism, which is the buildup of molecules to input ATP. With that in mind, the 2 processes we'll be discussing is cellular respiration, the usage of chemical compounds to harness energy from the environment, and photosynthesis, the uses of the sun to harness energy from the environment. The biggest difference between the two is cellular respiration is the breakdown of glucose, and photosynthesis is the making of glucose. Now, in the process of cellular respiration, it is a catabolic reaction that takes place converting fuel molecules such as carbohydrates, lipids, and proteins to release ATP molecules for work performance in a cell. It is also an oxidation-reduction reaction, or redox reaction, which is the storing or releasing of chemical energy. The equation for cellular respiration can be explained as glucose and oxygen yielding carbon dioxide, water, and energy. The oxidation or reducing agents are the glucose to carbon dioxide, and the reduction or oxidizing agents are oxygen and water. There are 4 stages in this process to break down, which we shall be talking about converting a single glucose molecule into 32 ATP molecules. Stage 1 is glycolysis, which literally means the splitting of sugar and occurs in the cytoplasm. Here is when glucose, the 6-carbon sugar, is split into 2 3-carbon molecules of pyruvate, a net total of 2 ATP molecules and 2 electron carriers NADH. This is an anaerobic reaction, meaning there is no oxygen required, though here it takes a little ATP action to get this process going, allowing glucose to be put in the form pyruvate for easier recognition in this task. The NADH is a coenzyme that carries electrons that will be helpful further along. Glycolysis is a series of 10 chemical reactions that can be divided into 3 phases. Phase 1, or steps 1 through 3, an inorganic reaction, converts glucose to fructose 1,6-biphosphates by adding 2 phosphate groups. Phosphofructokinase 1 controls a key step in reaction 3. This enzyme, PFK1, regulates glycolysis. When ATP levels are low, PFK1 is activated, allowing glycolysis to continue. When ATP levels are high, PFK1 is inhibited and glycolysis is slowed down. In phase 2, or steps 4 and 5, also known as the cleavage phase, this is where the actual splitting of glucose happens. The 6-carbon sugar is now broken into 2 3-carbon molecules which enter phase 3. In phase 3, or steps 6 through 10, also known as the payoff phase, ending not only the production of 2 pyruvate molecules but yielding 4 ATP and 2 NADH, noting that 2 ATP molecules were consumed through the making of pyruvate in phase 1, gives the net value of 2 ATPs in the final equation. Therefore, the equation for glycolysis is concluded as glucose, 2 NAD positive, 2 ATP, 2 inorganic phosphates yielding 2 pyruvates, 2 ATP, 2 NADH, 2 hydrogens, and 2 water molecules. As we move on into stage 2, the synthesis of acetyl coenzyme A, it can be called the transition stage because it is essential to connecting stage 1 to stage 3. It is also the first step in stage 3 known as the citric acid cycle, using itself as a substrate. The product pyruvate in stage 1 is transported from the cytosol to the mitochondrial matrix where it's oxidized into acetyl-CoA. The synthesis results in 1 carbon dioxide molecule and 1 NADH molecule per pyruvate, in which case there are 2 pyruvate yielding 2 carbon dioxide molecules and 2 NADH molecules, recalling the 2 pyruvate products from stage 1 are transferred into stage 2. Moving into stage 3, the citric acid cycle mentioned earlier, often known as the Krebs cycle, is where the fuel molecule pyruvate converted into acetyl-CoA's acetyl group is completely oxidized. We are still in the mitochondrial matrix, and this stage is 2 in anaerobic reaction. The 8 reactions start with oxaloacetate containing 4 carbon molecule reacting with the 2 carbon acetyl group to form a 6 carbon molecule citrate. Since this is a cycle, that means that the 2 additional carbon molecules have to be terminated before coming back around to the oxaloacetate. In reactions 3 and 4, this is where we lose the 2 additional carbon molecules and form 2 NADH molecules. In reaction 5, this is the only substrate-level phosphorylation generating the molecule GTP. The GTP transfers one phosphate into an ADP molecule to now make an ATP molecule. In phases 6 and 8, an additional NADH and FADH2 is produced. Just like the NADH molecule, FADH is just a coenzyme that will carry high-energy electrons to produce more ATP in the final stage 4. The cycle produces 3 molecules of NADH, 1 molecule of FADH2, and 1 ATP molecule per turn. Now summing up the Krebs cycle, we are ending with 2 molecules of ATP, 6 NADH, and 2 FADH because of the 2 acetyl-CoA entering from stage 2. In the final stage 4, we move out of the mitochondrial matrix into the inner membrane of the mitochondria. The 6 NADH and 2 FADH are ready to start donating their electrons. This generous donation of electrons passes through 4 protein complexes located in the inner mitochondrial membrane, creating a proton gradient. The proton gradient consists of 2 parts, a chemical gradient and the electrical gradient. The chemical is the difference in the concentration levels, and the electrical is the difference between the charges on each side of the membrane. Our focus will be on the electrical gradient. Electrons are passed from the electron donors to electron acceptors like oxygen. Oxygen in this stage is the final electron acceptor. The electrons enter the electron transport chain through complex 1 or 2. The electrons from the NADH are donated into complex 1, and the electrons from FADH is donated into complex 2. Coenzyme Q accepts the electrons from complex 1 and 2 and reduces into cytochrome C reductase. This transfers the electrons to complex 3 with lower energy. When this transfer is complete, complex 3 then transfers electrons to cytochrome C, and this reduces as well. It diffuses the membrane and moves on to complex 4, where the oxygen is reduced to form water. The transportation of electrons in complex 1, 3, and 4 create a proton gradient because they are the only complexes allowed to pump hydrogen into the intermembrane space of the mitochondria. Once the electrons have moved through the 4 complexes, ATP synthase converts the energy of the proton gradient into the energy of the ATP. ATP synthase is made up of 2 subunits, F0 and F1. F0 forms a channel allowing protons to flow through, causing a rotation. This rotation allows F1 to rotate as well. F1 is the unit that synthesizes ATP. So, the rotation caused in F1 allows the phosphate group to be added to the ADP molecule, making the remaining 28 ATP molecules for the total of 32 ATPs. Cells have so many ways to carry out the energy produced with oxygen, but how about without? When oxygen is not present, cells have other routes that they can take to make ATP. This is when the pyruvate from glycolysis is broken down and called fermentation. Organisms that produce ATP anaerobically have to take bigger quantities of fuel molecules to perform work. The 2 major types of fermentation are the lactic acid and the ethanol. Lactic acid occurs in animals and bacteria. Electrons from NADH are transferred to pyruvate to produce lactic acid and NAD+. Ethanol fermentation, or as most people know it, alcoholic fermentation, occurs in plants and fungi. Electrons from NADH are transferred to acetaldehyde to produce ethanol and NAD+. The acetaldehyde is when the pyruvate releases carbon dioxide. Although in both fermentations NADH is oxidized to NAD+, it is not shown in either equation because it's reduced in glycolysis. Whether cells go through an aerobic or anaerobic reaction, they make ATP, allowing work to be performed. The second way organisms harness energy from their environment is through the process of photosynthesis. Photosynthesis can be defined as a catabolic reaction because it is the building of carbohydrates from the usage of sunlight and carbon dioxide. Just like in cellular respiration, it is a redox reaction. The equation for photosynthesis can be explained as carbon dioxide and water yielding oxygen and glucose. The oxidation or reducing agents are water to oxygen, and the reduction or oxidizing agents are carbon dioxide to glucose. In photosynthesis, carbon dioxide molecules are reduced by oxidizing other molecules. Photosynthesis can be divided into two stages: the photosynthetic transport chain, also known as the light reaction, and the Calvin cycle, also known as the dark reaction, or the Krebs cycle. Since water is the ultimate electron donor, some organisms do not use water and therefore do not produce oxygen. Water is also abundant, making it harder to pull the electrons. So how do organisms get their energy this way? In the photosynthetic transport chain, there are protein pigment complexes called photosystems. These photosystems use the absorbed light energy to drive the redox reaction setting in the photosynthetic transport chain. The function of a photosystem is to absorb the light and convert it into energy used for the movement of electrons through the transport chain, so that it can be used to drive the synthesis of ATP and NADPH. First, we need to understand how light works to get the full understanding of light energy. Light is a type of electromagnetic radiation making its energy in the form of a wave. Visible light is the part of the spectrum that is used in photosynthesis. The wavelength of the visible light determines the energy absorbed. The absorption of the wavelengths is in the molecules called pigments. Chlorophyll is the major pigment in photosynthetic organisms which appear green. So why do we have colored flowers? These are called accessory pigments, allowing photosynthetic cells to appear other than green. The lack of absorbed light by chlorophyll are then filled by regions of the accessory pigment carotenoids. Carotenoids are fairly important in the photosynthetic electrotransport chain because it protects it from the damage. Although 95% of energy is given off as heat, a small amount is remitted as light. In the photosystems in thylakoid, the chlorophyll's absorbed light energy is transferred from chlorophyll molecule to chlorophyll molecule until transferred into the reaction center, converting light energy into the electron transport. When the reaction center is oxidized, the electron acceptor molecule is reduced. Now that we have an understanding of how light works in photosystems, it shall be easier to explain how the absorption of light energy is converted for the movement of electrons through the chain. This is taking place in the thylakoid membrane. The thylakoid membrane forms structures that resemble flattened sacs or pancakes called grana. When these pancakes stack, they are connected to one another by membrane bridges that also make it continuous. Although it may seem connected completely, the lumen keeps it separated as well. The area surrounding the thylakoid membrane is called the stroma. This is where carbohydrates synthesize and correspond with the cytoplasm. Photosynthetic cells also contain mitochondria, which allow them to form carbohydrates from carbon dioxide using mitochondrial respiration. The carbohydrates are then broken down to generate ATP. Two photosystems are needed to provide enough energy to pull electrons from water and then reduce them to NADPH. Photosystem 2 supplies electrons to the beginning of the electron transport chain and loses an electron, allowing electrons from water to be pulled. Plastoquinone carries the electrons to the next step. When the electrons are oxidized, they move into the cytochrome B6F complex, which will then in turn allow the electrons to pass through photosystem 2 into photosystem 1. This also acts as a proton pump, creating a proton gradient. Before the electrons can be moved from the cytochrome B6F complex to photosystem 1, plastocyanin has to carry and diffuse them through the thylakoid lumen. Following photosystem 1, the second input of light excites the electrons, allowing enough energy to reduce NADPH. This is known as the Z scheme because of the resemblance of the letter Z from the up and down movement. NADPH is formed when high energy electron is passed from photosystem 1 to a protein called ferredoxin. The enzyme ferredoxin NADP positive reductase then break down the formation to create NADPH. Too much of NADPH production could be harmful in the photosynthetic electron transport chain. To prevent this, electrons from photosystem 1 are redirected back into the chain. The alternative pathway is known as the cyclic electron transfer. Cyclic electron transport influences more ATP production because of the additional protons in the lutein from the excess high-energy electrons. Since the Calvin cycle also requires ATP, the accumulation of protons from the pump and the oxidation of water releasing protons and oxygen allow the use of the gradient to synthesize ATP molecules. Finally, ATP and NADPH are the energy sources needed to synthesize carbohydrates and using carbon dioxide in the process called the Calvin cycle. This process contains 15 chemical reactions that synthesize carbohydrates from carbon dioxide. It can be summarized into 3 main steps: carboxylation, reduction, and regeneration. The first step is the addition of carbon dioxide to a 5-carbon sugar named ribulose 1,5-biphosphate. It is catalyzed by the enzyme ribulose biphosphate carboxylase oxygenase, or Ribisco for short. Photosynthetic cells produce many of this enzyme. Ribisco and carbon dioxide diffuse into its active sites and produce a 6-carbon compound. The 6-carbon compound then breaks into 2 molecules of 3-phosphoglycerate and are considered the first stable products in the Calvin cycle. Since Ribisco is the key to add carbon atoms to form carbohydrates, carbon compounds must be reduced. NADPH from earlier, it then transfers energy and electrons to allow carbohydrates to synthesize from carbon dioxide. The reduction of the 2,3-phosphoglycerate goes through 2 steps. First, ATP is used to phosphorylate the 3-phosphoglycerates. Secondly, NADPH transfers 2 high-energy electrons to each of the phosphorylated 3-phosphoglycerates. Once this is done, energy transfer steps yield the formation of glyceraldehyde 3-phosphate, which is reversibly interconverted into dihydroxyacetone phosphate by the enzyme triose-phosphate isomerase. For every 6 triophosphates that are produced, only 1 exits. Lastly is the regeneration of the Ribisco. This is a 12-step process where 3 carbon compounds are reorganized and combined to produce Ribisco. ATP is required for this step. NADPH provides the energy incorporated into high-energy bonds for carbohydrate molecules, and ATP provides energy for the regeneration of Ribisco. The Calvin cycle is considered as a light-independent or dark reaction of photosynthesis. Both NADPH and ATP supply from the photosynthetic electron transport chain, where light is obtained and converted into chemical energy, making photosynthesis occur only in light. Since the cycle can make more carbohydrate than it needs or exports, it can upset the osmotic balance. So, in order not to mess with this balance, carbohydrates are converted into starch because they are not soluble. As we can see, every process is not 100% perfect. As we discussed earlier, excess light energy can be harmful to the cell. The other problem that may occur when NAD positive is cut short and NADPH is not produced fast enough is the light energy and electrons could be transferred into oxygen. Ribisco could add oxygen instead of carbon dioxide because of similar factors. When this enzyme adds an oxygen, it cannot be read by the Calvin cycle and releases carbon dioxide. The result is 1 molecule of 3 carbon atoms and 1 molecule of 2 carbon atoms called 2-phosphoglycylate. This process, when carbon dioxide is produced in light through oxidation, is referred to as photorespiration. This consumes ATP to convert the 2 carbon atoms into 3 carbon atoms that can be used. Photorespiration results in a net loss of energy from oxidation and carbon atoms from carbon dioxide using ATP. Though cold weather can reduce catalytic activity or the usage of pigments such as sildenafil to reduce excess light energy, mistakes can be made. So why does Ribisco have such low catalytic rates? It is actually to avoid the mix-up of carbon dioxide and oxygen, executing the idea of false production.